Hi everyone, welcome back to Neurobiology at Providence College. I'm Joe DeGiorgis. Today's lecture is part 2 on the paper titled A Novel Multigene Family May Encode Odorant Receptors, a Molecular Basis for Odor Recognition by Linda Buck and Richard Axel. It was published in Cell in 1991. And I mentioned earlier that Linda Buck and Richard Axel won the Nobel Prize for their work. So this is a very cool paper. Why don't we start by reviewing what we said in part 1 of the lecture. First of all, if we have a cell membrane with our phospholipid heads facing the aqueous environments like this, and this is the outside of the cell, and this is the inside of the cell. And we just represent the membrane like this. It turns out that there are a certain type of receptor known as G-protein coupled receptors. G-protein coupled receptors that are known to associate with G proteins. I'm going to draw it like this for now. And G proteins are proteins that bind to GTP, guanosine triphosphate. And there are 3 subunits of the G protein that are just named alpha, beta, and gamma, ABC, like this. And when the alpha subunit is associated with the receptor, it is bound to GDP, guanosine diphosphate. And when an odorant, in this case, this is their theory, that when an odorant or a ligand binds to the receptor, that it causes a conformational change, which is a change in the shape. And the change in shape is conferred through the membrane, that is, the entire protein here kind of changes its structure, and that causes the alpha subunit to drop the GDP and associate with GTP and gets released. So now we have the alpha subunit like this, and it's associated with GTP. Now the alpha subunit can associate with another transmembrane protein called adenylate cyclase. And when this alpha subunit binds to adenylate cyclase converts an ATP adenosine triphosphate into cyclic AMP. And then cyclic AMP goes downstream to affect another transmembrane protein, which is a cyclic AMP gated ion channel. So now you have cyclic AMP here, and that opens a door and allows ions from the outside of the cell to the inside of the cell, and this is sodium, and that depolarizes the cell. And the cells that we're talking about here are the specialized olfactory sensory cells, and they're a type of nerve cell. So if you depolarize the cell, you can cause an action potential, and somehow that action potential travels along the axon into the brain, where that nerve cell connects to other neurons in the brain and somehow causes us to sense smell, in this case. The alpha subunit then hydrolyzes the GTP to form GDP, and now we have an alpha subunit associated with GDP, which comes back and reassociates with the beta and gamma subunit and the receptor. So the system is reset for the next ligand to bind to the receptor. Now, there was some evidence that suggested to Buck and Axel that this was the mechanism that might be responsible for odor. One piece of information was that these specialized olfactory neurons are kind of shaped like this, and they have these cilia at their tips. And there's some support cells here in the tissue. This is within the nose, of course. And basal cells down here, some more support cells. But at any rate, it turns out that if these are removed, first of all, the organism can no longer smell. I mean, this was tested. And also, if you add an odorant to these cilia here in a petri dish, it turns out that there's cyclic AMP production, and it was known that G proteins trigger adenylate cyclase to create cyclic AMP. So it looked like this was the general mechanism through which we might detect odor, although that hadn't been proved. They say in their experimental strategy that they made 3 assumptions based on this early work. And the first assumption was that the receptors would bind G proteins. That is, they are G protein coupled receptors. They are G protein coupled receptors. That was their first assumption. Their second assumption was that they were members of a large family of proteins. That were similar, but each protein, each receptor, would bind to a different odorant. And the third assumption was that they would be expressed in the nose, because that's the organ we use to smell. So what they're trying to determine in this publication is what these receptor molecules are. Are they G-coupled protein receptors? Does a large family of them exist? And are they expressed in the nose? Okay, before we talk about the figures in the paper, we have to review what we know about DNA. First of all, we know that there are 4 molecules, 4 bases that make up DNA, and they refer to them by the first letter of their name, A, T, G, and C. And we know that they're held together like beads on a string. So it keeps going, and it's the order of the nucleotides that confers information. We also know that the DNA is double-stranded, and that for every T on, or every A on this strand, it is bound to a T on this strand. And for every T on the top strand, there's an A on the bottom strand. For every G, there's a C, and for every C, there's a G, right? And we know that this goes on on a chromosome, on a single chromosome, for a very long time. And we know that this twists. And the twist, of course, is the double helix, and Watson and Crick and Maurice Wilkins won the Nobel Prize for the discovery of the helix. And, of course, we also know that this very long double-stranded helix folds up to form a chromosome during mitosis and meiosis, the latter of which occurs during the formation of gametes, right, sperm and egg. And of course, we know in humans that there are 23 pairs of chromosomes. There's 22 autosomal chromosomes and 2 sex chromosomes, the X and the Y chromosome. And of course, if you're XX, you're female, and if you're XY, you're male, and you get one of each pair of chromosomes from your mother and father. So if this is chromosome 1 from your mom, and this is chromosome 1 from dad, then of course you have 22 more pairs of autosomal chromosomes, chromosome 2, 3, 4, all the way to 22. And then of course you have 2 special chromosomes that determine gender. You have an X chromosome and a Y chromosome, and if you're XY, obviously you're male, and if it's XX, you're female. I know this is all a review, but just stay with me. Okay, so now in the process of meiosis, there's a process of homologous recombination, and I'm going to shade in dad here, dad's chromosome. In the process of homologous recombination, there is a mixture of DNA from chromosome 1 from mom and chromosome 1 from dad. So you get this chimeric chromosome, something like this. And some of the chromosome, this newly formed chromosome, comes from dad, shaded here in black, and there are regions from mom. This chromosome shares DNA from both, and that gets packaged into sperm or egg. Of course, this process happens for all of the other pairs of chromosomes. There's some special issues related to sperm formation in terms of the XY chromosome, so let's leave that for now. The point is that the chromosomes that end up in sperm in males, of course, and eggs in females are a set of chimeric chromosomes, roughly half of which have DNA from the individual's mother and half of the DNA from the individual's father, or the grandparents of an individual created with that gamete. Okay, now let's say that there was a gene on some part of the chromosome, let's say right in here. Now we know that genes are parts of the DNA that ultimately encode for an RNA molecule, and in some cases, that is, if it encodes a messenger RNA, the messenger RNA encodes a protein. So genes in general are regions of DNA that encode for RNAs, and in most cases for proteins. So if we have a gene that encodes a protein, a receptor protein here, that is being conferred in this particular case, in this example, to this region here. But of course, we have 2 copies. I mean, this individual who has received this chromosome from mom and this chromosome from dad, this individual has 2 copies of the gene for that receptor molecule because they received 1 copy from their mom and 1 copy from their father. It's possible in the process of homologous recombination, in the formation of this chimeric chromosome, that there's an issue with homologous recombination. So we have a portion here, let's say from mom again, but it turns out that this region here is also from mom. That is, it didn't get processed correctly, so instead of getting this piece from mom, we ended up getting a bigger piece from mom, this whole part here. And then we still get this piece from dad. So now we have a piece here from dad, which is synonymous with this piece, so it's shaded. And then we get a piece from mom again, and a piece from dad again, okay, like this. But now we have this segment copied here, and this segment that contains a copy of the receptor molecule incorrectly spliced into this chromosome here. So now we have a copy of the gene from mom and a copy of the gene from dad in this chromosome. So in this way, this issue of homologous recombination created a duplication of the gene. We have a copy here and we have a copy here. And in fact, this region from dad might contain multiple genes, and same here. So you could be getting a whole region of multiple genes being copied. So you now have two copies of those genes on this chromosome that gets incorporated into a gamete and then becomes, part of the population. So of course, this is a form of genetic mutation, but just as long as the individual that has this can survive and reproduce, then it will be propagated in the population, and maybe it's totally innocuous. There's no problem at all. But my point is that now we have two copies of this gene that we said was a receptor molecule. And this would be two members of a family of genes now that are very similar to one another. I mean, they're identical when the problem happens, the duplication happens, they're identical. But over time, they can mutate and they can evolve to be expressed in different tissue types, or they could evolve to have completely different functions. Or perhaps they could be expressed in the same tissue type, but one binds to one odorant and the other binds to another. And over the course of evolution, you could have many gene duplication events, and the number of family members of those proteins could increase until you have hundreds of different receptor molecules, all slightly different from one another, and able to bind to a wide variety of odorants. Okay, so that's just an explanation of how you end up having large families of proteins that were derived from an ancestral gene and then come to generate dozens or even hundreds of family members. And it's not just receptors, it could be motor proteins like myosins and kinesins, which we're going to talk about in the next few days. But at any rate, it creates a large family of similar molecules. Now, if we go back to looking at the DNA as nucleotides on two strands, the top strand and the bottom strand here, and of course, they twist a little bit, that's the double helix, and these go on for a very long time. I mean, millions of base pairs in both directions. There are about 3 billion base pairs in one set of chromosomes. So there are two sets of the autosomal for 22 chromosomes and the X and the Y. So all total, there are about 6 billion base pairs. But at any rate, this is, of course, a base pair, and A and T is the base pair. So chromosomes, if you unravel them as two strands, it's a very long molecule. And I'm going to redraw this. Of course, you have two copies of this double-stranded molecule. You have, let's say this is chromosome 1. So you have a copy like this from your mother and a copy like this from your father. So I'm going to draw it without using any letters. I'm just going to draw two strands. Here's a strand, and here's a strand. And this is the 5' end of the top strand and the 3' end of the top strand. And this is the 3' end of the bottom strand and the 5' end of the bottom strand. And we said already that a gene is a region of the DNA that encodes for an RNA molecule, and those RNA molecules that are messenger RNAs, of course, get translated into amino acid sequence or proteins. They're read by the ribosome. Through transcription, an RNA molecule is produced that is an exact copy of one of the strands. And let's say it's this strand right here. This piece is a region that encodes an RNA molecule, and if it's a messenger RNA molecule, it gets transcribed into what they call heteronuclear RNA, like so. So it's identical, except that the T's in the DNA are now used, uracil. So at any rate, so you would have, for instance, an A, a U, a G, and a C, and so on. And it turns out that this hnRNA gets spliced. And there are some regions here that become part of the mature RNA molecule and some regions that get spliced out. So you might have a region here like this piece that is an exon, which becomes part of the mature RNA molecule. So we have this piece here. And then we have an intron, this region here, an intron, which gets removed. And then a second exon that gets spliced in next to this strand. So here's the break in between those two pieces. And then there could be another intervening sequence that gets spliced out and another exon, for instance. I mean, just as an example. So this would be exon 3. So that gets added on here to form this new RNA strand. And this is connected here, sorry. As one single strand of nucleotides, A, U, G, and C. Okay. Now it could be that there's alternative splicing. So this is exon 2 and this is exon 1 here. So you could actually have an RNA that's exon 1 and exon 2 only, or you could have exon 1 and exon 3 with 2 spliced out, or you could have exon 2 and exon 3 and so on. That's differential splicing. But at any rate, let's just say for the sake of our discussion that there are two introns, intron 2 and intron 1, and those two get removed during splicing, and the 3 exons get pieced together to form this mature RNA molecule, and then there's a poly A tail added and a 5' cap onto this end. And this is a mature messenger RNA. And we all know that messenger RNAs are those that get translated into proteins, into amino acid sequences. Okay, I'm going to redraw the RNA molecule. So we have a string of nucleotides, A, U, G, and C, and we have a poly A tail here. So I'm just not going to draw in the letters on this part of it, but there's a 5' cap, and this is the 5' end of the RNA, and this is the 3' end of the RNA, and we said that this is read by the ribosome. But it turns out the entire RNA is not translated into amino acid sequence. There's a point in here called the start site, and at the start site, the ribosome begins reading the nucleotides 3 at a time, and almost always the very first triplet that encodes an amino acid is A, U, G, which encodes the amino acid methionine. So it reads these 3, and then it reads 1, 2, 3, and there's another amino acid, 1, 2, 3, and another amino acid, and so on until it hits a stop codon, the triplet that encodes the stop. And then you end up with this amino acid sequence, and that's a functional protein. This region from the first nucleotide that encodes the triplet of the stop codon to the end is called the 3' untranslated region. The part that encodes the amino acids is called the coding sequence. And the part that is 5' to this AUG in this direction is the 5' untranslated region. And if you were to translate these regions, the 5' and 3' untranslated regions, you would find other stop codons, triplets that encode a stop, and the area from this stop codon to this stop codon is called the longest open reading frame. And we'll get to that in one of our laboratory exercises. At any rate, this coding sequence is important because that's the part that encodes the amino acid sequence, or is translated into amino acid sequence and forms the protein. Okay, if we now go back to our double-stranded DNA molecule, like this, I'm not going to draw the letters, but this is the 5' end of the top strand and the 3' end of the bottom strand, the 3' end of the top strand and the 5' end of the bottom strand. If we wanted to use the polymerase chain reaction, PCR, which you know is a technique to amplify a stretch of DNA, and we wanted to amplify a region that was a gene, that it encoded an RNA molecule that ultimately was translated into an amino acid sequence. Let's say we wanted to amplify something from here to here. We have to design two PCR primers, one that will bind to this bottom strand, and so it needs to be identical to the top strand because the top strand and the bottom strand complement one another. So you know, if this is a T down here, then this is an A up here. And if this is a G, this would be a C, and so on. So if you know the top strand, of course we know the bottom strand. But we need a piece of DNA that will bind to this T, C, let's just keep going a little bit. Here's a T, that would be an A, and so on. So we need about a 20 nucleotide stretch that will bind to this bottom strand, which is actually identical to these first 20 nucleotides on this top strand. So that PCR primer is very easy to design because you just start with this A and say A, G, T, and then whatever the remaining nucleotides are for about 18 to 22 nucleotides. And that's this, you can have it synthesized by a company, and they give you millions of copies of this PCR primer that will now bind to this region of the DNA. And then you need a second primer that is here, and it has to bind to this top strand. And that's easy to design too because it has to be identical to this bottom strand. So if this is a C up here, this has to be a G. And if this is a G, this has to be a C, and so on. And so this PCR primer binds to the top strand, and this PCR primer binds to the bottom strand, and then if you run the PCR reaction, you amplify millions of copies of DNA that are this long from here to here and are identical to this region of this chromosome. So to do the PCR reaction from genomic DNA, you would have to isolate some cells from the human body in this case, if we're doing humans, and you need to biochemically purify the genomic DNA, which is pretty simple. It takes about 45 minutes, and you can buy a kit to do that. But then in a test tube, you need, of course, you need the DNA, the genomic DNA. You need the primers, of course. which are these small stretches, this one here and this one here. You need extra nucleotides. You need A's, T's, C's, and G's to synthesize new DNA molecules. You need DNA polymerase, and you usually need some magnesium, and you need some buffers that are in there. And you all know that in the PCR reaction, the machine heats the DNA up to separate the two strands. It cools it to allow the primers to anneal or to bind to the DNA, and then it warms it up to somewhere around 72 degrees for elongation, that is for copying the DNA. And I'm not going to go through the PCR reaction. You can find that online if you want to review that topic. Okay, so that's how you would PCR amplify a segment of genomic DNA, but you could also amplify the RNA molecules, messenger RNA molecules. And to do that, you have to biochemically purify the RNA molecules, which have, of course, been spliced, and poly A tail has been added, and the 5' cap, and so on. And this is the 5' end of the RNA and the 3' end of the RNA. But in order to do PCR from RNA molecules, you have to convert it into DNA. So you can buy a string of T's, and the T's, of course, bind the poly A tail here, and then you use a chemical, a molecule, a protein called reverse transcriptase, and it reads this nucleotide up here. So if this is a G, it adds a C, and if this is a C, it adds a G, and so on. So it reads the top strand, and it makes a complementary strand to the top strand, except when this was done, you added A, T, C's, and G's. So now this is a DNA strand, because as we know, this top strand is an RNA molecule, so it's composed of A's and U's, not T's, and C's, and G's. Then you destroy this RNA up here by adding an RNase, and RNase chews up the RNA, like that. And now we have the bottom strand here with our GC, GC, and our T's and so on. And when you add DNA polymerase, it reads the nucleotides on the bottom strand now, and it makes a complementary strand of the bottom strand. And so this top strand is identical to the original RNA molecule, except the U's have been now replaced by T's, and this is now a double-stranded DNA molecule. They call this copy DNA or cDNA. That is reverse transcribed messenger RNA that's been converted into a double-stranded DNA molecule. Now, just like we did here with our making primers that would bind to this genomic DNA, you can make a primer here that would bind to the bottom strand of this DNA molecule, and one that would bind to this top strand of the RNA molecule, and it would amplify a region, including the PCR primers, but also the nucleotides that are between them. You'd get many copies of this stretch of this RNA molecule. Now that's cool because you could be PCR amplifying the region of the RNA that encodes for the protein, the coding sequence, and you don't have to worry about introns that might be found when trying to PCR amplify genomic DNA. Okay, you guys, 2 days of lecturing and still we haven't gotten to the first figure of the paper. So one more time, let's say that we have a piece of DNA and now chromosome 1. And of course, there's 2 copies of chromosome 1, one from mom and one from dad, but they're extremely similar. And in fact, many of the genes on mom's chromosome 1 and dad's chromosome 1s are absolutely 100% identical. And sometimes there's mutations. So of course there's slight genetic differences between human beings. And those differences make us different, first of all, but could also be used to determine maternity. It could be used for crime scene analysis. I mean, those slight little differences in the nucleotide sequence can be detected through PCR and other approaches. And at any rate, we're just going to look at one copy of chromosome 1, let's say from mom. So this is chromosome 1 from mom. And let's just realize that there's a chromosome 1 from dad, but it's very, very similar to chromosome 1 from mom. So now if we had a region of the DNA that encoded a receptor molecule, let's say here to here, and that created, of course, an RNA molecule with a poly A tail, and that made a receptor molecule, a protein, say 1. If there was a gene duplication event, now you would have 2 genes for receptor molecules, and when the gene duplication event happened, the 2 genes would be absolutely identical. But over time, point mutations and other more egregious mutations can happen between these 2 receptors, and so they start to differ a little bit in terms of their nucleotide sequence, and ultimately that can be conferred to the amino acid sequence. That is, you have point mutations in the DNA that result in mutations, single amino acid substitutions, single changes between, of course, the RNA molecules, the 2 RNA molecules, and the 2 receptors. So you have receptor 1 and receptor 2, and they're still really, really closely related to one another, and their sequences are very highly similar, but there are some differences. And it could be that over time, those 2 receptors could be expressed in different tissue types or at different stages of life and so on, or they could be expressed at the same time and in the same tissue, but along with changes in sequence, you could have changes in function. And so one possibility is that when they change function, in this case, talking about odor, they change what types of odorants can bind to receptor 1 versus receptor 2 and trigger that cascade that we worked on with the G proteins. Then, if you had another gene duplication event, so these are slightly different from one another now, and then at some point there's another gene duplication event, so you have another receptor. Now these, if this one was the one that was duplicated, these are identical again at the time that the duplication event happened, but then these 2 start to mutate away from each other, and now we end up with 3 different receptor molecules that have 3 different functions, or in terms of our conversation of odor, this detects odor number 1, odorant number 2, and odorant number 3, something like that. And you could have many more receptors that detect dozens or hundreds of different odors, right? Now, since each one of these is making a unique RNA molecule, if you purified the RNA, you would purify the RNA from all 3 genes. And if there were, dozens of genes now for receptors, then you'd end up having dozens of RNA molecules in a test tube. So you biochemically purify RNAs, in this case from the tissue in the nose, because they're trying to figure out what the receptors are in the nose. Then now you have all of these RNA strands. Of course, you're isolating all of the RNA strands, all of the poly A RNAs, so it's not only for receptors, but for every other type of protein as well. But they're all in this test tube, and you can convert them all into copy DNA. Each strand gets converted into double-stranded DNA, and now you can do PCR. But it turns out that if you have a PCR primer, let's say, there's going to be a primer that binds here and a primer that binds here. If the sequences are similar enough between the gene of receptor 1 and receptor 2, and receptor C, then the same primers might be able to bind to this region here too, like this. I mean, if this we were going to amplify from genomic DNA, but I'm just saying that the primers could bind to all three receptor molecules at the genomic level, but they can also bind to all three copy DNAs. So there's copy DNA from receptor 1, copy DNA from receptor 2, and receptor 3, right? And the copy DNA, again, is the RNAs, the transcribed RNAs from the genes that are being expressed in that particular tissue, and it's turned into double-stranded DNA. Now, if we do a PCR reaction with these cDNAs, these pool all of these cDNAs, not just the cDNAs of the receptors, but all of the cDNAs created from that tissue type, then the primer sets could bind potentially to all of the different genes or all of the different RNAs that are translated into receptors. So if we draw this, and this is the cDNA of number 1, this is the cDNA of number 2, and this is the cDNA of number 3. Then the primers can bind here and here on the first, here and here on the second, and here and here on the third. And oftentimes you get point mutations, but oftentimes you get point mutations between the RNAs, but you don't get major shifts in length. I mean, it's possible to have a change in length, but oftentimes when you look at families of proteins or families of genes, the size tends to be very similar with just small changes in nucleotide sequence and ultimately amino acid sequence. So now if you amplify this region of this gene, this region of this gene, this region of this gene, or cDNA rather, and you ran it out in a gel, all three bands would be the same size. So you'd have one that's this size here, same size here, and same size here. And if you load this into a gel like you've done in Gen Bio, you load it into the well here, and then the bands run down and you get a band. And this band right here looks like a single PCR product, but it's actually a mixture, could be a mixture of this, this, and this, and they all run together in the same band because they're all the same length. They're all the same molecular weight.